minutes later. And the same tone's going to play and the light's going to flash again. Okay, there it goes, right now. And now you can see, just 10 minutes into the experiment, that we've equipped the brain by photoactivating this area to overcome the expression of the sphere memory. Now, over the last couple of years, we've gone back to the tree of life because we wanted to find ways to turn circuits in the brain off. If we could do that, this could be extremely powerful. If you can delete cells just for a few milliseconds or seconds, you can figure out what necessary role they play in the circuits in which they're embedded. And we've now surveyed organisms from all over the tree of life, every kingdom of life except for animals we see somewhat differently. And we found all sorts of molecules. They're called halorhodopsins and archirhodopsins, which respond to green and yellow light, and they do the opposite thing of the molecule I told you about before, the blue light activator, chenorodopsin. Let's give an example of where we think this is going to go. Consider, for example, a condition like epilepsy, where the brain is overactive. Now, if drugs fail in epileptic treatment, one of the strategies is to remove part of the brain, but that's obviously irreversible and there could be side effects. What if we could just turn off that brain for the brief amount of time until the seizure dies away and cause the brain to be restored to its initial state, sort of like a dynamical system that's being coaxed down into a stable state? This animation just tries to explain this concept where we've made these cells sensitive to being turned off with light, and we beam light in, and just for the time it takes to shut down a seizure, we're hoping to be able to turn it off. And so we don't have data to show you on this front, but we're very excited about this. Now, I want to close on one story which we think is another possibility, which is that maybe these molecules, if you can do ultra-precise control, could be used in the brain itself to make a new kind of prosthetic, an optical prosthetic. I already told you that electrical stimulators are not uncommon. 75,000 people have Parkinson's deep brain stimulators implanted. Maybe 100,000 people have cochlear implants, which allow them to hear. There's another thing, which is you've got to get these genes into cells. And new hope in gene therapy has been developed because there are viruses, like the adeno-associated virus, which probably most of us around this room have, and it doesn't have any symptoms, which have been used in hundreds of patients to deliver genes into the brain or the body. And so far, there have not been serious adverse events associated with the virus. There's one last elephant in the room, the proteins themselves, which come from algae and bacteria and funguses and all over the tree of life. Most of us don't have funguses or algae in our brains, so what is our brain going to do if we put that in? Are the cells going to tolerate it? Will the immune system react? And it's early days. These have not been done in humans yet, but we're working on a variety of studies to try and examine this. And so far, we haven't seen overt reactions of any severity to these molecules or to these, the illumination of the brain with light. So it's early days to be up front, but we're excited about it. I wanted to close in one story which we think could potentially be a clinical application. Now, there are many forms of blindness where the photoreceptors, our light sensors that are in the back of our eye, are gone. And the retina, of course, is a complex structure. Let's zoom in on it here so we can kind of see it in more detail. The photoreceptor cells are shown here at the top, and then the signals that are detected by the photoreceptors are transformed through various computations until finally that layer of cells at the bottom, the ganglion cells, relay the information to the brain, where we see that as perception. In many forms of blindness, like retinitis pigmentosa or macular degeneration, the photoreceptor cells have atrophied or been destroyed. Now, how could you repair this? It's not even clear that a drug could cause this to be restored because there's nothing for the drug to bind to. On the other hand, light can still get into the eye, right? The eye is still transparent, and you can get light in. So what if we could just take these chenorodopsins and other molecules and install them on some of these other spared cells and convert them into little cameras? And because there's so many of these cells in the eye, potentially they could be very high-resolution cameras. So this is some work that we're doing. It's being both led by one of our collaborators, Alan Horsager at USC, and being sought to be commercialized by a startup company, EOS Neuroscience, which is funded by the NIH. And what you see here is a mouse trying to solve a maze. It's a 6